Today, we are diving into something absolutely mind-blowing. We walk around every single day thinking of ourselves as just one unified individual, right? But the truth? You're actually a bustling, highly communicative, incredibly complex, microscopic society. Every single function of your body and ultimately your overall health depends entirely on trillions of individual cells working together in absolute perfect harmony. Okay, let's dive right into this, because the source material highlights this truly beautiful concept. Cells must demonstrate a chemical fondness for other cells. I just love that phrasing, don't you? Chemical fondness. It really reveals the ultimate stakes of our biology. The cells in your body are constantly talking to each other, listening, collaborating. When they maintain this fondness, you thrive. But when this microscopic social network breaks down, when that cellular crosstalk stops, cells become isolated, vulnerable to injury, and that's when disease takes over. So here's our quick roadmap for this explainer. We'll hit the cellular society, what's inside the eukaryotic cell, the plasma membrane, protein folding and stress, and finally, how cells stay connected. Let's kick things off with Section 1, the cellular society, looking at prokaryotes and eukaryotes. Before cells even begin to figure out their specific day jobs, life basically divides into two main camps. You've got your prokaryotes and your eukaryotes. Prokaryotes, like bacteria, well, they're the simple core subjects of molecular biology. They don't have a distinct nucleus or specialized organelles. But human bodies, we are made of eukaryotes. Eukaryotic cells are larger. They have this highly organized, complex anatomy, and they feature a very characteristic set of membrane-bound compartments called organelles. They also have these proteins called histones that bind with their DNA, which is a critical feature for keeping all that complex genetic code organized. Now, because eukaryotic cells are so advanced, they go through a process called differentiation, which allows them to specialize. And they specialize into 8 chief functions. We've got movement for generating force, conductivity for nerves, there's metabolic absorption, secretion, excretion, and respiration, plus reproduction. But above all, and this is absolutely crucial, communication. Communication is literally the key for them to survive as a dynamic, living society. Moving right along to Section 2, inside the eukaryotic cell, a cellular metropolis. And this brilliantly illustrates the sheer complexity we're talking about here. It's a fluid-filled metropolis. That fluid space is the cytoplasm. And far from being just, empty water, it is an incredibly crowded, bustling space that actually takes up half the cell's volume. It's packed with thousands of enzymes involved in intermediate metabolism and crammed with ribosomes working tirelessly to synthesize proteins. Suspended right in the middle of this space are the cell's organs, the organelles. Right smack in the center of this metropolis is the nucleus, the cell's largest membrane-bound organelle. It's enclosed by a nuclear envelope that is pockmarked with all these little pits, or pores, that let chemical messages flow in and out. The nucleus is essentially the control center for your genetic information. Now, the DNA chain in eukaryotic cells is incredibly long and super fragile. To keep it from breaking, the nucleus uses those histones we mentioned earlier to carefully fold the DNA into tightly coiled chromosomes, kind of like wrapping thread around a spool so it doesn't tangle. Just outside the nucleus, we find the mitochondria. You've probably heard them called the power plants of the cell, and for sure, that's exactly what they are. Through a really complex process called oxidative phosphorylation, they take in nutrients and oxygen to generate adenosine triphosphate, or ATP. ATP is basically the fundamental currency of cellular energy. It fuels all the complex respiration and metabolism required to keep this cellular city running day and night. Which brings us to Section 3, the plasma membrane, or as I like to think of it, border control. So the actual wall of the cell acts a lot like a strict microscopic nightclub bouncer. Its basic structure is a lipid bilayer, and it has this brilliantly clever amphiphilic design, which really just means it has 2 totally incompatible solubilities. The round heads are hydrophilic, meaning they love water, and the tails are hydrophobic, meaning they absolutely hate it. Because of this, the membrane spontaneously organizes itself into 2 protective layers, shielding those tails from water. This creates a highly effective barrier that only lets certain lipid-soluble molecules, things like oxygen and carbon dioxide, pass through easily. Now, what's really interesting about this slide is how the cell solves the huge problem of getting necessary things through that tough lipid wall. It uses proteins. Proteins embedded in the membrane perform most of the specific tasks. On one hand, they act as physical transport gates, channels, and pumps for electrolytes and amino acids. On the other hand, they act as cellular receptors, poking out into the extracellular space to listen for chemical messages, specific molecules called ligands. When a ligand fits into a receptor, kind of like a key into a lock, it instantly changes the cell's behavior. Let's get into Section 4, protein folding and stress. Basically, cellular maintenance. Because proteins are the ultimate workhorses doing all the heavy lifting, the cell has to maintain a strict system of quality control, known as proteostasis. This is a vital state of cellular balance. It ensures that proteins are synthesized at the right speed, folded correctly, and degraded when they're just no longer useful. It's a highly adaptable system that relies on ribosomes to make them, chaperones to help fold them, and disposal systems like lysosomes to break them down. To actually work, a protein must physically transform. When it's first translated from RNA, a protein is basically just an unfolded polypeptide, a floppy random coil of amino acids. Inside the endoplasmic reticulum, or ER, it has to fold perfectly into a highly complex three-dimensional native structure. Only when it has this precise 3D shape can it actually do its job as a receptor or a transport channel. So, here is the crucial point. What happens when this incredibly delicate process fails? Viral infections, environmental toxins, or even just a massive cellular workload can stress the endoplasmic reticulum, leading to a traffic jam of misfolded proteins. If the cell's protective response can't keep up, these abnormal proteins start clumping together. And this microbiological failure, it has massive, real-world consequences for us. Unresolved ER stress and misfolded proteins are directly linked to devastating conditions like Alzheimer's disease, Parkinson's disease, and diabetes. Finally, let's zoom back out for Section 5, how cells stay connected, the biological glue. Let's just think about this for a second. Cells are incredibly small, they're squishy, and as we just saw, they're enclosed only by a flimsy fluid lipid membrane. So how on earth do they bind together robust enough to form a muscle strong enough to let you pick up a textbook? How do they form these durable, tough tissues? Well, the answer to that is the extracellular matrix, or ECM. Local cells called fibroblasts secrete this intricate meshwork of fibrous structural proteins, like strong collagen and rubber-like elastin. The ECM acts as a complex biological glue, a gel-like ground substance that surrounds the cells. Think of the cells sitting inside this tough matrix kind of like raisins suspended in a pudding. It doesn't just hold them together, it also provides a pathway for nutrients and waste to diffuse. But the cells don't just float near each other. They actually physically hook together at specialized regions of their plasma membranes called cell junctions. Tight junctions hold cells together so tightly they form a literal leak-proof seal. Adherents and desmosomes provide incredibly strong mechanical attachments, bracing the cells against pulling forces. And gap junctions? They act as communicating tunnels. This is vital. For example, gap junctions permit electrical action potentials to spread rapidly from cell to cell, which is exactly how your individual heart muscle cells synchronize to beat perfectly as one. Which really brings us full circle. From the intricate folding of proteins in the endoplasmic reticulum to the electrical pulses flashing through gap junctions, your entire existence relies on microscopic teamwork. When they maintain their chemical fondness, you experience health. When they don't, disease takes root. So, having seen the staggering complexity of this cellular city, I want to leave you with this question.